Let us see the nucleotide excision repair pathway of UV-induced damage. So UV irradiated E. coli can regain their ability to survive after incubation in dark, although we know that these UV irradiated E. coli can survive if they kept in light, because in light they can photo repair their DNA. But even if they are kept in dark, they can regain their ability to survive. So, however, they recover more slowly than when incubated in light. So, their recovery was a bit slow as compared to recovery in the light. This observation suggests that the bacteria use some process other than photoreactivation to repair the UV-induced DNA damage. So there must be some mechanism which repairs the DNA in dark. Richard Satlow and William Carrier, and working independently, two other scientists, Richard Boyce and Paul Howard Flanders, they used a similar approach to investigate this alternative process in 1964. Both groups cultured E. coli in the presence of H3-thymine. This is labeled thymine, and they cultured bacteria in this to label the DNA, and then irradiated the cells with UV light to induce the formation of cyclobutane thymine dimers. Then they carried out number of steps. For example, first of all, they incubated the UV irradiated cells in the dark so that photoreactivation could not take place. Secondly, they removed samples after various incubation times and added TCA, trichloroacetic acid, to them. Then they separated acid insoluble DNA from acid soluble nucleotides. So two types of products were present. One is acid insoluble DNA and the other one is acid soluble nucleotides. In the next step, they digested the DNA and oligonucleotides to release intact thymine cyclobutane dimers. In the fifth step, and finally, they detected the released dimers by chromatography. The experiments revealed as the incubation time in the dark increases, cyclobutane thymine dimers disappear from the acid insoluble DNA and appear in the acid soluble oligonucleotide fragments. So, as I mentioned earlier, there were two products. One was the acid insoluble DNA, intact DNA. The other one was the acid soluble oligonucleotides. So, they separated these two products and then digested these products and then analyzed where the cyclobutane thymine dimers are present. So they observed that as the time was increased in dark, incubation time was increased in dark, the thymine dimers, cyclobutane thymine dimers were increased in oligonucleotides, and they decreased in the intact DNA. These results were correctly interpreted to mean that the bacteria can excise an oligonucleotide containing a lesion and replace the excised oligonucleotides with newly synthesized and correct DNA. Subsequent studies showed that eukaryotes and the archaea also have nucleotide excision repair pathways as they are present in bacteria.